Hi everyone, welcome to Group 4's presentation on transmissible cancers. My name is Gabi and I will be introducing the project and some background information on cancer. First of all, we wanted to address why our topic is relevant. Many, if not all of you, have heard of cancer, a common disease which is one of the leading causes of death in the world. Many of us know someone affected by it. Cancer affects most animal species and it relates to the cell cycle, evolution, and ecology, which are fundamental aspects of biology that we are all very familiar with. Cancer has been a major topic of research for centuries, yet there is still very little that is known about both cancer and transmissible cancers. Research regarding transmissible cancers may lead to new discoveries about cancer in general. So what is cancer? Cancer is the name given to a collection of related diseases. In all types of cancer, some cells in the body divide without stopping and spread into nearby tissues. As we learned earlier, our cells grow and divide to form new cells through a process called the cell cycle. As our cells age and become damaged, they die and new cells take their place. However, there are times when the growth of cells is unrestricted, which is when cancer can appear. As cells become abnormal, some of them survive when they should die, and new cells are formed unnecessarily. When extra cells grow without restriction, tumors can form. Solid tumors are masses of tissues which can form as a result of cancer. Other types of cancer, such as in the blood, usually do not form solid tumors. There are two kinds of tumors which you may have heard of, malignant and benign. Malignant tumors are cancerous and can spread to surrounding tissues, create tumors elsewhere, and can grow back when removed. On the other hand, benign tumors do not spread, and when they are removed, they usually don't go back. Most benign tumors aren't life-threatening, with the exception of benign brain tumors. A reason for which cancer cells do not act the same way as normal cells is because they are not as specialized, and they are able to ignore important signals from the body, as well as fool the immune system into thinking they are regular cells. Cancer is caused by changes that occur in our genes that control the way that our cells function, specifically how they divide and grow, which Ellie will elaborate on in a few minutes.
Hi, my name is Inaya, and I'm going to be giving some background information on transmissible cancers. A transmissible cancer is a tumor that spreads directly from one individual to another by the transfer of cells between them. Species that may carry transmissible cancers include Tasmanian devils, soft-shell clams, and canines. Transmissible cancers are clonal, meaning that the cells originally came from the mitotic division of one somatic cell. The fitness of these cells is defined by the ability to be transmitted between hosts. The following factors have been identified as essential in order for cancer to spread. 1. The tissues of the tumor must encourage shedding of many malignant cells. 2. The environment of the new host must allow the invasion of these foreign cancer cells in order for them to survive. And 3. The tumor must have sufficient cell plasticity, which is the occurrence of molecular and phenotypic changes. This is required so the cells can adapt in response to environmental changes of a new host. Barriers that can prevent the transmission of cancers are ones that don't conform to these requirements. Luckily, excluding organ and tissue transplants, there haven't been any cases of naturally occurring transmissible cancers recorded in humans. This is largely because of our strong immunologic responses. There must also be a route of transmission for the tumor cells to travel from one host to another, such as physical contact. Additionally, transmissible cancers are likely able to thrive in their new host as a result of low genetic diversity in the species.
Hello, I'm Yimin. I will talk about bivalve leukemia-like transmissible cancer. In humans, cancer called leukemia infects blood-forming tissues like the bone marrow and lymphatic system. However, leukemia-like cancer called hemic neoplasia has been found in multiple marine species, including marine bivalve species such as clams, mussels, and oysters. The leukemia-like disease has been found in bivalves across hundreds of kilometers on the east coast of North America. Despite being so far apart, the hemic neoplasia cells were nearly genetically identical. Transmissible cancer in softshell clams has been documented since the 1970s, indicating that it is at least 40 years old and possibly much older. It is unclear how the cancer cells spread, but clams filter feed, raising the possibility that leukemia spreads through clams' filtration of seawater contaminated with neoplasia cells. Studies have found that blood cells from a leukemic clam could survive in natural seawater conditions for more than 6 hours with minimal cell death, suggesting that ocean current is a plausible route for cancer cell transmission from one clam to another.
Hi, I'm Hannah, and I'll be talking about Tasmanian devil facial tumor disease. Despite Tasmanian devil facial tumor disease, DFTD, only being first observed in 1996, the epidemic has skyrocketed and become a serious threat to the Tasmanian devil population, threatening the population with extinction. The disease is spread by transporting living cancer cells from one Tasmanian devil typically through biting. This means that the tumors grow in and around the mouths of Tasmanian devils, which comes with its own set of challenges. The cancerous infections are constant without seasonal variants nor sexual selection of the disease, and it's known that both DTFD and canine transmissible venereal tumor disease have evolved that allows the major histocompatibility complex, the MHC, to not recognize them as foreign cells. The MHC has molecules that are within the membranes of every cell in the body, identifying the cells as belonging to the body. Somehow DFTD has evaded this system, therefore not attacked by the immune system, as the cells are not recognized as foreign. It was recently discovered that there are two versions of transmissible cancers within the Tasmanian devil population, DFT1 and DFT2, both being entirely separate from one another. DFT1 is believed to have originated in female Tasmanian devils, while DFT2 carries a Y chromosome, meaning it is most certainly originated in male Tasmanian devils. The fatality rate of Tasmanian devils that are infected are almost 100%. The average amount of time between noticing a Tasmanian devil is infected and the time that they pass away from the condition is approximately 6 to 12 months.
Hi, I'm Max, and I'll be talking about canine transmissible venereal tumor. This is a cancer that affects dogs and other canine species, primarily on the male and female genitalia, and in some cases the nasal cavity. It is transmitted between hosts through coitus and other common social canine behaviors, such as sniffing, licking, or biting during fights. CTVT is spread through the transfer of cancerous cells from subject to subject, caused by the exfoliation and transplantation of cancerous cells during sexual intercourse or other social contact. Intact neoplasm cells are transplanted into the skin or mucosa of dogs, where they begin to grow and spread. Significant structural differences exist between the cells of this tumor and normal dog cells, including highly specific chromosome aberrations. For unknown reasons, the body does not attack these foreign cells, which allows them to grow and spread unchecked. One challenge faced in the fight against CTVT is the prevalence of wild and stray dog populations in some parts of the world. CTVT has been largely eliminated in Europe and North America due to the population control of stray animals. However, CTVT remains endemic in other parts of the world due to the unchecked populations of strays and the inability to treat these animals.
My name is Ellie, and I will be talking about transmissible cancers from the genetics and cell cycle perspective. Mitosis is the most common form of cell division. It is the process where replicated chromosomes separate into two new nuclei. Mitosis occurs in most cells and is a process that happens countless times daily. Before mitosis occurs, the cell undergoes three important phases: the G1 phase, S phase, and G2 phase. Before each phase, there is a checkpoint where the cell ensures that there are no errors in organelle replication or DNA replication. If a cell skips a checkpoint and the work done in the phase prior is incomplete or has errors, mutations are caused in the DNA. Almost all cancers are the result of gene mutations. Oftentimes, these mutations cause malfunctioning genes, which cause malfunctioning cell proteins, and ultimately, malfunctioning cells. One example of a harmful mutation in transmissible cancers causes the formation of a malfunctioning p53 protein. The protein p53 is a tumor suppressor protein whose function is to regulate the cell cycle and keep cells from proliferating uncontrollably. The second example of a harmful mutation in transmissible cancers causes the formation of a malfunctioning BCL2 protein, which regulates cell apoptosis. Transmissible cancer cells evade apoptosis, and one of the main causes is a mutation in the anti-apoptotic BCL2 protein.
Hey everyone, I'm Ali Tufan, and I'm going to be covering transmissible cancers in an evolutionary concept. Devil facial tumor diseases. Examination of karyotypes based on Taylor et al. Reveals the existence of at least 4 major chromosomally distinct strains of DFTs. The occurrence of multiple cytogenetic strains show that DFT is an evolving disease with genetic variants that are likely to have different morphological, physiological, or other biological characteristics. Canine transmissible venereal tumor. High chromosomal stability of CTVT cells indicates that this cell lineage has well adapted to its niche and most likely has reached the final stages of stepwise evolution. The analysis based on Mergia et al. Indicates that the tumor arose between 200 and 2500 years ago. However, since CTVT represents the oldest known mammalian somatic cell line, in the same article it is also stated that time span that was mentioned earlier most likely represents a later bottleneck effect that CTVT gone through. Bivalve transmissible neoplasia has been widely described since the 1960s from various mollusks such as clams, mussels, oysters, and cockles across the world. The neoplastic mollusk cells are characterized by higher than normal DNA content and aneuploid karyotypes, indicating that the cells most likely have undergone major chromosomal rearrangements in their evolutionary phase.
This is Max again, and we'll be talking about treatments for transmissible cancers and upcoming research in the field. Canine transmissible venereal tumor is the most treatable type of transmissible cancer that has been extensively researched in the 20th century. There are a number of different treatment methods. First, surgically removing the tumors has been shown to be an effective treatment. However, the cancer relapses in 30% of surgeries due to the exfoliation of cancer cells. Next, irradiation has been shown to effectively shrink these tumors, but prohibitive costs prevent this from becoming widely used. Finally, chemotherapy has been accepted as the treatment of choice for CTVT. Vincristine injections are affordable and widely accessible, making them the premier option. Research into devil facial tumor disease is still developing. It was only identified as a transmissible cancer in 2006. Since the disease is still so new, no firm treatments have been determined yet, although measures have been taken to protect Tasmanian devils. First, researchers have brought 2 unaffected devil populations to mainland Australia for quarantine. Next, a few individuals have been found who show resistance. There is hope that through natural selection, these disease-resistant individuals will breed on their own and eventually become the dominant variety of devils. Bivalve transmissible neoplasia requires no physical contact for transmission and has been found to spread between populations that are hundreds of miles apart. Currently, there are no known treatments for this cancer, but there's ongoing research to understand how the cancer cells emerge and escape its host. Researchers are also trying to determine an origin for this cancer, which will help to better understand how the disease is spread and find preventative measures of treatment.
Here are the sources we've used to conduct our research. For the additional portion of our project, we've developed an 8-day lesson plan to educate students about transmissible cancers. This class, the first one, will focus on cancer in general. Students will have a short discussion in breakout rooms about the basics of the cell cycle, and will then have a class-wide discussion on what they know about cancer and how we can relate this previous knowledge to the cell cycle. There will then be a short lecture focusing on the relevance of cancer, what cancer is, and the cell cycle and cancer.
The second class will be focused on introducing transmissibility of cancers. First, we'll reflect on what students learned about cancers last class and make predictions about transmissible cancers. Following the lecture, students should be able to complete a worksheet explaining what transmissible cancers are and what factors make it more likely for cancer to successfully transmit.
On the third day, we will learn about transmissible cancers among marine bivalves. To begin with, we will review transmissible cancers' characteristics. After that, we will introduce neoplasia cancer cells in bivalves and their mechanics. Third, we will have a group discussion on the challenges that marine bivalves are facing. By the end of the day, students should be able to explain why transmissible cancer is a challenge to marine bivalves and what are the spreading mechanics of neoplasia cancer cells.
On the fourth day, we're going to have a 10-minute brainstorm on how we think Tasmanian devil facial tumor disease spread from one to the next. Then we'll have a 20-minute lecture about how it's dangerous, the life expectancy in the 2 different types, and compare them. Then we're going to contrast how it's different from neoplasia cancer, and then we're going to have a breakout group for 5 minutes to discuss the learning goals.
This day will focus on CTVT. First, we'll review the past day's materials on the background of transmissible cancers. Next, we'll have a lecture on the unique elements and biology of CTVT. Then the class will discuss potential research in the field. By the end of the lecture, students should have an understanding of the biology, history, and unique properties of CTVT.
On day 6, the class will learn about transmissible cancers from a genetics perspective. First, students will do a review of the previous lessons. Second, students will get into groups and make a poster on mitosis in the pre-mitotic stages. Lastly, there will be a lecture on mutations. By the end of this class, students should be able to describe mitosis in the pre-mitotic stages, while additionally being able to understand what mutations are and how they can cause cancer.
For day 7, our plan is to cover transmissible cancers in an evolutionary concept. First of all, we'll be doing a recap of what we learned from previous days, and then we'll dive deep with combining our knowledge from previous days with the concept of evolution. For this class, you should be mainly focusing on learning at least one evolutionary concept that plays a critical role on evolution of transmissible cancers.
Day 8 will focus on the treatments and upcoming research of transmissible cancers. First, we'll recap the similarities and differences between the different types. Next, we'll discuss methods of treatments and methods to prevent the spread of cancers. By the end of the lesson, students should have a clear understanding of the treatments of transmissible cancers and methods of preservation.
By the end of the lessons, students should be able to complete a quiz highlighting the important information from the lessons. We've attached a link to this quiz to test your understanding of transmissible cancers. Thank you for listening to our presentation.